award ceremony for the Michael John Page Postdoctoral Fellowship Award. This was started in 2013 and is in honor of a graduate student who used to be in my lab. And for those that don't know me, I'm Ross McGilvray, and I used to be a researcher in this building at one time. So I just wanted to say a few words about Mike. We started this award because he was tragically taken from us at age 36. And so the Page family, the Center for Blood Research and the Department of Biochemistry and Molecular Biology all put together a fund which initially covered the costs, but more recently, Roger Page, who's Mike's father, has actually endowed this award. So it'll be here for longer than I will. So just to give a brief history, Mike was brought up in Thunder Bay, which is on the very west end of Lake Superior, a very cold place. He graduated from high school where his father was actually the physics and chemistry teacher for grade 11 and 12. So not surprisingly, he went on to do a scientific bachelor's degree at Carleton University. He then came to my lab in 1999 and graduated in 2004 with his PhD. Mike was a very, very bright guy, very independent. He sort of taught himself how to do X-ray crystallography, working with Natalie, and ended up, the majority of his work was mutating a bacterial protease to try and see if we could convert it to a mammalian thrombin-like enzyme. So he was very successful in his studies here. He was able to get his own studentship from Canadian Blood Services. He also was very good at training and mentoring other students in the lab. And Mark Bleakley was a guy who came in not knowing what to do as a third-year undergraduate summer student. And by the end of his interactions with Mike, he was a fully blown graduate student by day, but he was also a bouncer at Dentries by night and excelled at both positions. The one thing about Mike that made him such fun was that he really enjoyed life, be it our Friday afternoon beer sessions, watching his beloved Vancouver Canucks, who back then were not very good, and looking after his dog. So he just had a really great life. After my lab, he went to work in Washington University in St. Louis, working with Enrico de Serra, who's a very prominent thrombin biochemist, and is known to be somewhat of a hard taskmaster. So for Enrico to say such wonderful words about Mike, such as publishing 8 papers in 5 years, some of the best work from his lab. He got his own fellowship from the American Heart Association. And as Enrico said, Mike had ingenuity, deep knowledge, and then again, that enthusiasm for science. So having worked with Enrico, Mike then moved to San Francisco to work with Charlie Craik, who's this gentleman here. And Mike went as a sort of junior faculty member under Charlie Craik's mentorship. When he was there, he developed a new area for him, which was these restricted interaction peptides, which were comprised of a marker, a hydrophobic region, and then a targeting region for a peptide. And the idea was these peptides could be used to localize specific protease activity inside a cell, which could then be viewed non-invasively by MRI. The idea is that the peptide is cleaved at the protease recognition site, and then the hydrophobic part of the peptide inserts into a local membrane, which you can then visualize. So this has resulted in a patent. Mike formed a spin-off company called BioPaint. Everything was going really well for Mike. And then unfortunately, at the end of work on a Friday, he suddenly passed away eating dinner at age 36, probably from atrial fibrillation. So to honor his name and memory, we set up this award in Mike's honor. Initially, the Center for Blood Research and BMB were very generous in supporting this. And then many of you will have seen Roger Page, who has been to every one of these, except for the pandemic ones, and who I hope is viewing it on Zoom. Although he called me half an hour ago and said he was having problems, so we'll see. Anyway, with that introduction, I'd like to ask Philip van Petegem to invite or to introduce this year's winner.
All right, well, it's my pleasure to introduce Seby Chen, who's a postdoc in the lab. So I'll just give you a little bit of scientific background about Seby. So Seby performed his undergrad studies at McGill University in biochemistry. He also stayed there for his PhD. He joined the lab of Kalle Gehring, and just like Michael Page, he actually was exposed to crystallography there. He solved some very nice structures of magnesium transporters, actually, pretty hard class of molecules to crystallize. He got a doctoral research award also from the FRQS over there, and published, a bunch of really nice papers during his PhD. And then since the summer of 2021, he joined my lab here at UBC, and he started working on, a pretty exciting protein, at least from our viewpoint, the ranitidine receptor, which is a very large membrane protein. And he'll, probably will talk about this protein later on. Seby actually amassed a ton of data, actually, and I would say in the years that he has been here. So he recently actually got a very nice paper reporting the first neuronal type 3 ranitidine receptor, but he has a lot of papers and I think actually a backlog of structural data that all needs to get published. So I think if I can characterize Seby, he has an incredible work ethic. But at the same time, I would say Seby is also an extremely helpful person in the lab. As many members of the lab can definitely attest to, he's kind of the person that can really show people from the very basics onwards how to process cryo-EM data, how to go through that, and actually is very patient and very diligent in guiding people in that process. And other than that, I would say Seby is also incredibly, of course, he's very patient, works very hard, but at the same time, he's incredibly driven. He picks up new methods very fast. I mean, structural biology is definitely by no means an easy task. But one of the examples I can give, there's a method called planar lipid bilayer electrophysiology, where you can measure single channel currents on ranitidine receptors. It's notoriously hard. This is only a few people actually have, I would say, the mental characteristics to sit down patiently, wait for these bilayer recordings to happen. There's a lot of failure. And I would say Seby is one of the few people actually that is able to do that. And I think, it's a great characteristic. And other than that, Seby is a great lab member to have. I have very, lots of high level interactions with Seby about general projects. We don't get bogged down in technical details too much because he really always has very great ideas. And I won't diverge on his personal life because I think he's going to mention that himself, but I can tell he's also been very active in the EDI committee, and he takes, something he values also a whole lot is actually mentoring students. And with further ado, I'd like to invite Seby and tell us a little bit about his work and his life.
disconnect. Oops. See my mouse. Okay. Hi, everyone. My name is Seby. First off, I'd like to thank Philip for the great introduction, and also the Page family and Department of Biochemistry and Center for Blood Research for awarding me this award this year. I really appreciate the concept behind this award that encourages postdocs to be well-rounded in that they not only should have pursue academic excellence in research, but also have activities outside research for a fulfilling life. So I'm asked to give a 25-minute presentation about what I did for my research and also a little bit about what I do outside research. So I'll be giving you a brief view of my PhD and also postdoctoral work today. But before I begin, I'd just like to acknowledge that the land on which we are gathered here today is the traditional, ancestral, and unceded territory of the Musqueam people. Okay, so first off, what do I do as a researcher? I'm a protein biochemist and a structural biologist. As we know, protein are the molecular machineries that basically does makes us what humans and does everything inside the body. And that is from the DNA, which gets translated to amino acids, which then folds into these three-dimensional structures. There are proteins, which gives its function. But when there are mutations in the gene, then it can sometimes be deleterious, and that could lead to a protein malfunction, and that can lead to diseases. So as our job as a protein biochemist is to express and purify these disease-relevant proteins and try to understand what is their normal function and what happens when they malfunction, and study them with biophysical assays. And also as a structural biologist, we determine their three-dimensional structures because by looking at their structures, we then can understand what are the changes that occur when it undergoes its normal function, what are the conformational changes, what are the active sites, how does it bind its ligand, and how can we design drugs to target them for therapeutic purposes, for example. So there are main two methods for structural determination for these days. One is the X-ray crystallography, which has been mentioned. So basically you purify the proteins and you make these beautiful protein crystals, which then you shoot intense X-rays at the synchrotron, for example, to get diffraction, and then using gathering these information using computational softwares, you can get structure factors and get electron density mapping, which you can build your atomic model and analyze your protein. The second method, so that X-ray crystallography is what I did during my PhD. And the second method is called electron microscopy, which is what I am doing here at UBC. Similarly, you put purified proteins in a cryo-EM grid, which is like super tiny grid, which then you plunge rapidly into liquid ethane into a very thin layer of ice, which then you shoot electrons with this added with this electron microscope, which then allows you to take cryo-EM images of these particles, which then you average them together and using computational software to generate a 3D volume, which you can also build your atomic model. So when I got this award, I did a little bit of background research on what Dr. Page has worked on during his graduate studies and also postdocs. So it appears that our path is quite similar in that we're both a protein biochemist and a structural biologist. So as mentioned, he worked on these proteases that from bacteria, and then he was trying to do some protein engineering to alter its substrate specificity, for example. And then he was also using X-ray crystallography to determine crystal structures of these mutant proteins and try to understand how are they altering their functions from a structural point of view. So unlike Dr. Page, I was more interested in membrane proteins instead of enzymes. So as we know, membrane proteins are membrane-embedded proteins, which are important for a wide variety of functions inside the cells. Some of them are shown here. For example, they're involved in transport, signal transduction, and cell-to-cell adhesion, for example. So for me, I am interested in the proteins that are involved in transporting ions or substrates across the lipid bilayer. So for transport proteins, there are different classes, mainly separated into channels and transporters. So the channels, the distinction between channels is that when the protein ion channel opens, there is a continuous conduction pathway that allows the pathway to be open to both sides. Whereas for transporters, there are many flavors of them. On the other hand, only when it's active, only one cavity is exposed to one side. So there is never a continuous pathway across the two sides. So for my PhD work at McGill under Dr. Calagaring's lab, I was working on this class of antiporter, which transports ion in one direction, but at the same time transport another ion in another opposite direction down its concentration gradient. So it's a form of active transport. So that's what I'll be talking about first in like three short stories about what I did. So this transporter is called CNNM. It is a magnesium transporter. So its name is CBS paired domain divalent metal cation transport mediator. So in human, there are 4 members. CNM2 and CN4 have been proposed to have efflux, magnesium efflux activity. So they sit at the large intestine or renal epithelial cells, where at the apical membrane, there are transporters or ion channels that will uptake magnesium from the food or the urine. But CNMs will be at the basolateral membrane, where it's involved in extruding the magnesium back into the blood. So it's important for the uptake of these magnesium, which is an essential ion. So as we can understand, when these proteins are mutation occur in these proteins, they will cause some genetic diseases. One of the most known for CNM2 example, for example, is hypomagnesemia. So it is what happens when you have low blood serum magnesium level, and you can get seizures, and in severe cases, you can have mental retardation or brain malformations. And for CNM4, the well-known more associated disease is called Jalili syndrome. It's a rare disease in which you have problem with your enamel formation and also some problem with the cone receptors in your eyes. So you get bad eyesight and also bad teeth. So on top of that, the CNM proteins have been implicated in cancer because there have been regulator proteins that will regulate this protein that will inhibit its magnesium efflux activity. So there will be accumulation of magnesium inside the cells, and that is said to be favorable for the tumor to proliferate because magnesium is often bound to ATP, which is associated with energy status and energy metabolism and protein synthesis inside the cell. So by regulating this channel, the cancer cells is more prone to become tumors and metastasize. So I would just be giving you 3 short stories about what I did in my PhD to characterize structurally about this protein. So this protein, it's a domain architecture. In the N-terminus, there's an extracellular domain, followed by a transmembrane domain called domain of unknown function 21, because nobody really knows what it does. And then there's a soluble fragment, which composes 2 domains. One is the CBS pair domain, which is has been the only domain known structurally prior to my PhD, which shown as a dimer, and it binds magnesium ATP in the middle, where it's able to dimerize and sends magnesium ATP in that fashion. So it's sort of serving like a magnesium ATP sensor. And there's this loop that will interact with regulatory proteins. And followed by this is a cyclic nucleotide binding homology domain. So it doesn't really much known about it other than sequence similarity to cyclic nucleotide binding domains. So the first story when I first entered PhD was to first characterize this soluble domain that hasn't been characterized before. So like many do, we purify the proteins and then try to crystallize, get these crystals and diffract them. And ultimately we're able to determine the crystal structure of the CMBH domain from 2 members of the CNM family. So it looks like a canonical cyclic nucleotide binding domain. However, we did a lot of biochemical assays to confirm whether it indeed binds cyclic nucleotide, but it doesn't. Because looking at more structurally, overlaying with some of the canonical cyclic nucleotide binding domains, it seems to have residues that are physically clashing into the cyclic nucleotide, the putative binding site, and also it's lacking some extra helices that are involved. And on top of that, instead of using this pocket to bind cyclic nucleotide, it actually uses it to dimerize. So it's, again, occluded in this dimerization phase interface. So for us, it's interesting to see how nature has evolved this cyclic nucleotide binding domain into a different function, which is dimerize. And to take a step further, we then see what is the effect of deleting this domain whether it's essential. So there's this cell-based assay that we can measure the transport activity of magnesium transporter. So it's seen that when we delete this protein, it no longer has this efflux activity. And also when we mutate the dimerization interface, it also removes its activity. So suggesting it's an important regulatory domain. So with that, we added additional piece of the puzzle to this understanding the CNM protein function. And with that, we like to solve a bigger piece of the puzzle in the second story, where I solve a larger piece, which is the whole cytosolic domain, including the previous CBS pair domain and CMBH domain in two different conditions in unligated and magnesium ATP bound states. So this now allows us to observe these different conformational states upon magnesium ATP binding. For example, the CBS pair domain in the unligated form, it's this wide twisted state. Upon binding magnesium ATP, and now it's a flat piece dimer. And in the C-terminal domain, the CMBH domain also have different dimeric configuration upon. So it sort of serves as a hinge-like domain that's try to keep these CBS pair domain together. So we also identified the magnesium ATP binding site and characterized some of the residues involved in binding these magnesium APT, because some of these residues are actually disease relevant, mutated in the Gillies syndrome or hypomagnesemia patients. So a lot of these mutations, when we deleted them or mutated them, we tested the affinity for magnesium ATP by isothermal titration calorimetry, for example. They really abolished the binding. So in order to connect what is the relevance of this magnesium ATP binding and its protein function, we also performed the functional efflux assay again. We found that those mutations that cause abolishment of magnesium ATP binding also abolish efflux activity, suggesting that this CPS pair domain is an important magnesium sensing domain, that it's important for regulating the channel function. But as we know, this is a membrane protein. We're interested in the business end, which is the transmembrane domain, which back then we did not have AlphaFold, so we can't really predict that. So we had to do this experimentally. So of course, we tried to express the full-length human or the mammalian CNN proteins, but to no avail. Like we couldn't get enough protein, or it's just not well behaving. But thanks to the observation that this protein is actually highly conserved in all species. So in addition to existing in vertebrates and humans, but they also exist all the way down to fungi, chloroplasts, bacteria, and plants. So it's very universally conserved. In the eukaryotic CNM have the 4 domains I mentioned before. In the prokaryotic ones, they lack the extracellular domain. And instead of the CMBH domain, it has another core C domain. But the central region, which is the transmembrane domain, CBS pair domain, is highly conserved. So with that, we went with plan B, which is to try to maybe work with the easier proteins from bacteria, because they're easier to express and purify. And from that, we can also gain structural insights. With these structural insights, we can then help us understand more about the human protein. And there's also like the debate on whether they are actually membrane transporters themselves, because some people are questioning with just as a dimer 6 transmembrane helices, whether they are able to transport magnesium themselves, or they're just regulating other proteins. So with that, I ordered about 9 different thermophilic bacteria, CNNMs, because thermophilic is easier, more stable protein, so it'd be more chance of crystallizing them. And then I was not in my PhD lab, I didn't really have much experience working with membrane proteins and high throughput screening. So I was at a conference, and then I talked to one of the speakers who's presenting on that. And then she actually invited me to go to her lab to do an exchange. So with the fellowship I got from the PhD, I also did a 3-month exchange. So she's located at Structural Genomic Consortium at Oxford University. So she's Dr. Katarina Dürer, so she had her lab. And there I did like small scale screening of these different proteins. So for each one, you have to make different constructs for crystallization. And also because it's membrane protein, you don't really know what kind of detergent you would like. So you need to really screen them to see which detergent your protein is happy in. So not only need to get a band on the gel, but also have to have homogeneous dispersion on the gel size solution, for example. So long story short, some of them were able to express well, but they didn't crystallize. But ultimately, after many optimization and everything, I was able to obtain the crystal structure of one of the prokaryotic CNNM, which is actually from a thermophilic archaea, which is like derived from a wastewater from a nuclear power plant or something. And this is the crystal that grew. It's like a flower petal-like crystals, which was able to determine two different conformations of this construct, which contains the conserved region, which has the transmembrane domain and the CBS pair domain. So as we can see, this is all before AlphaFold, just right before AlphaFold came out. So it's a novel transmembrane fold. For the first time, we see that this is able to form this transporter fold with an inward facing cavity in which there is a negative charged cavity here. It has a reentrant helix that forms like a belt around the three transmembrane helices. And inside this cavity, there is actually a density that corresponds to magnesium, in which is coordinated by various conserved residues that is conserved all the way down to a human. And interestingly, there's a pi helical term that coordinates that. And because of having the two conformations, we are able to make some sort of movie, allowing us to propose a mechanism of how this CBS pair domain acts as a regulatory domain, in which that under low magnesium levels, it exhibit in an inhibited state, and under high magnesium ATP levels, then it's in an activated state in which the channel can then do its transporter function. And because of the debate about whether it's actually a transporter itself, I also devised an in vitro assay, that transporter, liposomal transport assay using purified protein to establish that indeed this protein has transport function on its own, because previous assays were done in cells, so it wasn't sure if it's the direct or indirect transport activity. So next, I would like to move into my postdoctoral work, which is now focused more on this other type of membrane protein, which is the ion channel at Philip Van Petegen's lab here at UBC. So here I work with a class of protein called ranitidine receptor, which is involved in muscle contraction in the process known as excitation contraction coupling. So that's how your neurons go to your send signal to your muscle, depolarize the membrane and activate calcium channel, and then which the calcium then binds to ranitidine receptor, causing it to release more calcium, and that's how you get muscle contraction to occur. In the human, there are three isoforms of LYR, and LYR1 is mostly found in the skeletal muscle. So some of the disease relevance would be myopathies, like central cord disease, for example. Our LYR2 is more found in the heart, so it's implicating a lot of the cardiovascular diseases, such as CPVTs, type of ventricular tachycardia. And then LYR3 is the more found in the brain, so there are some disease relevance to like epilepsy, for example. So again, I'll just go into like 3 short stories about some of these work that I've done and contributed in my postdoc. The first story is the story that I collaborated with Omid Haji Gassami. He was a previous postdoc in the lab. He's now a professor at University of Calgary. So for this one, we were interested in understanding the regulation of scorpion toxin, which is known to bind ranitidine receptor and induce subconductant states, has been shown a long time ago. But it was unclear how exactly where it binds the receptor and how exactly does it induce the subconductant states. So we determined the crime structures of these ranitidine receptor 1 in complex with the impaired calcin, the scorpion toxin, found that it binds to these region that's just right above the transmembrane domain. It sort of acts as a plug that like plugs the channel, but it's not a perfect plug because there are still some gaps that allows the calcium to conduct through a leak a little bit. So as mentioned, I was also contributed to this paper by doing some planar lipid bilayer experiments where we purify LYR proteins, reconstitute into liposome, proteoliposome, put it into this and reconstitute it into this planar lipid bilayer and try to measure single channel activity of ranitidine receptor. So this is an example of what happens of one of the recordings. So in the closed state and the inflection going up is when it opens. So the calcium will generate some current. So an example of a real-time measurement of the single channel activity, it goes really fast, like it goes up, that means the channel is open, for example. And it's like going to a burst mode where like it's going crazy, for example. So by adding the impaired calcium, we're able to observe the effect of impaired calcium on ranitidine receptor in that it doesn't plug it completely. It induces the subconductant states that's sort of leaky that still allows a little bit of calcium to leak through, but it's like sometimes very prolonged. So this is the work that we've done here in this one. And then the second story I want to talk about is also on the skeletal muscle isoform or androgen receptor 1. So RWR1 is also implicated in a disease called malignant hyperthermia. So it's a rare disease in which it requires a genetic mutation, but also a triggering agent. In a lot of cases, these triggering agents are volatile anesthetics that are used in surgeries. So what happens when you have mutations in RWR1, this is a mouse model for one of the disease mutant, you will get an elevated body temperature and then you will die if you don't get promptly treated by one of the the only FDA approved drug so far, which is dantrolene. So it's a rare adverse effect of the anesthetics, but it's fatal. So we're trying to understand what is the mechanism behind this disease, malignant hyperthermia causing mutation. So we obtained some of these homozygous mice from the collaborator, and we purified mutant and also wild type androgen receptor 1 and tried to understand the structure and function relationship by solving cryo structures. So we're able to determine that. We found that indeed the mutant proteins actually exhibit more open channels under the same conditions, supporting the fact that it is a gain of function mutation. And the mutation is located in the transmembrane domain here in the linker between S4 and S5 linker. So this view from the top, so it's between the S4 and S5, so it's here. So when the channel opens, it will sort of be in contact with the S6 helix. So this is looking from the side view. So in this residue, when the channel is closed, it doesn't really do much. It's like it's not contacting anything, residues, it's exposed to the solvent, but in the open state is now contacting this S6 helix, now forming this interaction. So when we mutate this threonine to a more hydrophobic isoleucine, now it forms a more favorable interaction with this hydrophobic pocket. So what we believe the mechanism here is that there are some mutations that will disrupt the closed state, and then therefore you get more open channels. But in this case, it's opposite, is that you're stabilizing the open state. So therefore the end result is the same. You get more frequent opening of RWR1 and therefore higher sensitivity to triggering agents, for example. And we were interested in understanding whether we can reverse the effect of this gain of function mutation with dantrolene, which is the FDA approved drug. We added and then we determined cryo structures. We weren't able to observe significant differences in the particle distributions, but we were able to observe that these dantrolene is bound to this peripheral region that is co-binding with ATP. So this is still work in progress. And the last one is about the story on RWR3, which is one of the last members of RWR, which doesn't really have much structural information because the other previous two members, there's a lot of crime structures. These RWR3 is very outdated, like 20 years ago, for example, and it's really low resolution. And this paper was just published last week. So we investigated the effect of the structure of RWR3. So RWR3 is in the brain, is in the hippocampus neuron. It's the only isoform expressed in the dendritic spine. So it's been proposed to be the player that's first signaled the calcium transduction, followed by the other members. So we're able to determine the structure of RWR3 in different conformational states. And again, we're able to observe different conformational change upon channel opening, similarly here at the pore. And we're able to observe the canonical binding sites of the known canonical ligands. But in addition, we are able to observe some novel ligand binding sites in the N-terminal disease hotspot, where there is a chloride ion and also a ATP molecule bound to this domain. So with that structure, we're able to now map some of the disease-associated mutations. And we found that actually around like 75% of the mutations that are disease-associated in the ClinVar database is associated with the disease called epileptic encephalopathies, which is a severe form of epilepsy. So we found that a lot of mutations are now clustered in this novel binding site for the chloride ion ATP binding site, which we characterize. So it's interesting to sort of connect this link between the structure observation and also the clinical observations. With that, I would like to move on to talk about the activities I do outside the lab. So I have been a postdoctoral member for 3 years on the departmental BMB EDI committee. It's 3 years, so my term has finished this year with these lovely people that are in the department. Here we discuss the EDI related issues, try to make the department better in terms of EDI. And with that, and also in addition to that, we also plan a lot of EDI focused events. For example, for me, I was in the team that initiated and co-led the BNB mentorship program. So it's a program that we initiated all with some of the training members here. And we basically, the idea is we pair up mentees, student trainees in the department and help them gain some interactions, because especially during the COVID, there's a complaint of lack of community across the department. And also as an immigrant myself, and also the only person in my family that did science, I felt when I was navigating academia, there was a lot of hardship, like trying to understand what's going on in academia. So we felt the initiative was good to help some of the trainees who might were in my similar shoes as me, to maybe gain more insights about like how to navigate the academia or just find research positions or whatnot, for example. So in addition to that, we plan some sort of group events that's, for example, this virtual careers night, in which we invited 6 panelists from different diverse backgrounds who were all graduates of BNB, and then came back to talk about what they're doing now. Some of them are in academia, industry, or even a lawyer, for example. And then we also plan social events where we, because, you know, as I said, because of COVID, people really need a way to connect, especially for postdocs. Like grad students, you have classes and everything, but postdocs, we're just like sequestered to our lab. We don't really have much interaction with the other labs. So these helps us to connect with the other labs. Science outreach, so a lot of EDI event from the EDI committee. We also did science outreach. So for example, this year we did the science rendezvous, where we had a lot of stations that teach young kids about like science we do, like for example, colorimetry, how to isolate DNA from strawberry and whatnot. For me, I handled this station. I also designed this station, which is like basically grew some lysozyme crystals, and then I showed the kids what they look like under microscopes, and then kind of talk to them about why are we interested in these crystals, and why are we interested in protein structures. So just trying to get some science into these kids. And then on top of that, outside the UBC, I was also signed up to do some science outreach through Skype a Scientist, which is a website which you can sign up, and then they'll match you with classrooms across the world. So for me, I signed up, and then I was matched to a grade 11 AP chemistry class from Maricosta High School, which is located in California. So there I just Zoomed with them, talked about what I do as a protein biochemist, structural biologist, answer some of their questions, and hopefully inspire some of them to be scientists themselves. And then on top of that, we do a lot of mentoring and teaching. As I said, I wasn't like in science background before. I was very fortunate to have a lot of great mentors. Dr. Garen, for example, who took me in when I was a clueless undergrad, and the PhD student that was supervising me, he taught me everything, like, for example, how to even use a micropipette, for example. So there were a lot of mentors, excellent mentors in my career. So I'm always happy to supervise students, mentor grad students, even from other labs. And I also participate in the program, BNB mentorship programs, in which I mentor 2 graduate and 1 undergraduate students. I was very happy to help one of the undergrad to obtain land a research position to help him like draft the cold emails to the professor, for example. And I also volunteer to teach undergraduate course, for example, about 404, which is really fun. And then outside academic round, I was into hiking recently. So these are some of the pictures I got from hiking. Yeah, so like one funny story is like, this is when I was climbing the Chief, and it's like 20 minutes in to the hike, was with some of the lab members. And then I was like feeling dizzy, and I was almost fainted. Yeah, because it was probably because of my first hike. And also I was too afraid to ask them to stop. So then I learned a great lesson is I shouldn't really care about what other people think. If I really need to rest or take it slowly, I should just say that, because or bad things like this happen. But thankfully, a young lady was really nice who came down the trail, saw me really pale, and was like, "Oh, do you need help?" So she gave me a candy, and then I pretty much recovered right away. But then I thought about, oh, should I just give up and go home instead? But then being a scientist in me who doesn't like to give up, I like decided that I drove all the way there. And then so I like continue on thinking that I'll at least reach the first peak. So this is us at the first peak. And then we ended up going to the second and third peak. And these are other ones that we did, which is higher elevation or longer. But I really encourage people to do hiking, especially in Vancouver. It's like very nice nature that we have here. And it's really a good way to unplug yourself because there's virtually no cell reception in some of the mountains. And it really teaches you how to have perseverance, because a lot of times I like questioning my life about my life decision, like why am I on this long hike? But a lot of times it's very similar to my scientific career. Like a lot of times I'm questioning my scientific career decisions. But then at the end, if you persevere through enough, you will then also reach the mountain and you will enjoy the beautiful view. Yeah, so lastly, I'd like to thank everyone who has helped me out during my PhD and postdoc, the funding sources, and again, the Michael Page Endowment, CRB, and BNB for giving me this award. Yeah, and lastly, for everyone who came here to listen to my talk. Thank you.